The title for today's topic is Types of Cell Deaths. There are as many as 10 to the power 9 cells in an adult human which die every single day. The NCCD, that is the Nomenclature Committee on Cell Death, has devised a classification mechanism for classifying the types of cell deaths. And this classification encompasses all morphological, biochemical, and functional aspects. And according to NCCD, there has been 3 types of cell deaths which has been classified so far. Okay, so the type 1 cell death, which is known as the apoptosis, is a programmed cell death. The type 2, which is called as autophagy or self-demise, and the type 3, which is the necrosis or the accidental cell death. So each of these types, they are characterized by certain features, certain changes, including morphological, biochemical, or molecular changes. For example, if we see the apoptosis, it is characterized by the shrinkage in the cell and the formation of apoptotic bodies without the leakage of the contents in the vicinity. The type 2, which is the autophagy, it is characterized by the appearance of the vacuoles, autophagosomes, and these autophagosomes, they fuse with the lysosome, which ends in the organelle digestion. The third type, which is the necrosis. Necrosis involves basically swelling of the cytosol and the organelles, and is accompanied by the breakdown of plasma membrane and the leakage of cellular contents, which is absent, like the leakage we don't see in the apoptosis. So the normal cells, they are directed to different types of cell deaths depending upon the several mechanisms as well as several factors. So if we look at the morphological changes, because morphological change in the cell is the most important thing by which we can classify the type of cell deaths. So if we see the morphological changes in apoptosis, as I already said, it is accompanied by the shrinkage and the condensation of the chromatin, which results in the, which is followed by the fragmentation and formation of blebs. Now, the formation of blebs results in the ultimately in the formation of apoptotic bodies, which are taken up by the phagocytic cells. The necrosis, on the other hand, it is characterized not by shrinkage, but by the swelling of the organelles, and necrosis is also characterized by the breakdown of the membrane and the release of the cell content. And the autophagy. So autophagy, basically it involves the formation of the vacuoles and the formation of the result autophagosome, which then lyses with the lysosome and results in the digestion of a particular organelle or cell. Now, if we, if we compare the necrosis, apoptosis, and autophagy, we see that in necrosis there is in necrosis, the damage, it is produced by some external agents like infection or physical, mechanical assault to the cells, and which causes the rupturing of the cells and the leakage into the surrounding tissues. So when there is leakage from the cell into the surrounding tissues, and non-specific immune response is elicited, which leads to the local inflammation. But in apoptosis, apoptosis is basically a programmed, controlled type of cell death, which involves the various pathways. There is an intricate relationship between various mechanisms or various pathways which control this apoptosis. So cells are destroyed in a manner which is called as autodestruction, and in apoptosis there is no leakage of the contents into the surrounding tissues, such that no immune response is elicited and there is no collateral damage to the surrounding cells. Now, coming to the autophagy. So autophagy is characterized by lack of chromatin condensation, which is a feature of apoptosis. There is massive vacuolization of the cytoplasm, and double membrane autophagic vacuoles accumulate. Now, the thing about autophagy is that autophagy is also a process by which cellular organelles, they are devoured by lysosomes to clear away unnecessary or dysfunctional components. So it is a way which can promote both cell survival as well as cell death depending upon the circumstances. we will we will be focusing more on the apoptosis in this series of lectures. Now, why apoptosis, which is the programmed cell death? So apoptosis is basically a physiological phenomenon which has many functions. For example, if we consider the the stages of the embryonic development, we see that apoptosis plays an important role in what is called as tissue sculpting. For example, the digits in our hands and feet, the digits, they are attached during the initial phases of the embryonic development, and apoptosis leads to the elimination of certain cells, which leads to the digitization of the appendages. So it is an important phenomenon, physiological phenomenon by which tissue sculpting jo hai, that happens. Apart from that, we might have seen the life cycle of a frog, wherein you see the tadpole, wherein tadpole is converted into an adult frog. So the tadpole loses its tail. The loss of tail in the tadpole is also an example of the apoptosis. Similarly, we can see the apoptosis happening at various physiological phenomenon, like the development of neurons. So earlier on, the nerve cells that are produced, they innervate the peripheral organs. So only the nerve cells that reach their target organs survive, whereas the nerve cells which fail to reach the target organs are undergo apoptotic or programmed cell death. So basically, apoptosis, it is a quality control process by which abnormal, superfluous or non-functional as well as potentially dangerous cells like cancerous cells are eliminated. Also, it aids in the sculpting and tissue homeostasis. Now, the causes of apoptosis. So we can have both physiological as well as pathological causes for apoptosis. Physiological, we already enumerated few, like embryogenesis and fetal development, and regression of lactating breast after weaning. And similarly, we have the formation of when we have the formation of T lymphocyte cells. So the T lymphocyte cells are get matured into the lymphocytes by a mechanism called negative selection. So the negative selection is based upon the apoptosis or programmed cell death. Now, coming to the pathological. So what are the pathological features which lead to the programmed cell death or apoptosis? So the major being the damage to the DNA due to radiation or any chemical agents, because DNA, it is we don't want the genetic blueprint to be destroyed or altered in an individual. And second is the accumulation of misfolded proteins. For example, we have Lewy bodies which get accumulated, leading to stress or apoptosis. This happens in diseases like neurodegenerative diseases like Alzheimer's diseases or Parkinson diseases. Also, the cell death in viral infections and organ atrophy contribute to the pathological apoptosis. So there are certain morphological and biochemical changes that take place in the cells undergoing programmed cell death. So the first change that is to be seen in this process is the shrinkage and condensation. So shrinkage and condensation is the key feature of the cell undergoing apoptosis, followed by cytoskeleton collapse, disassembly of nuclear envelope and nuclear chromatin condensation, DNA fragmentation. Again, blebs. Blebs is a key feature by which apoptotic cell is recognized, formation of apoptotic bodies, and certain phospholipids are exposed at the cells which serve as the signals for phagocytosis, and leakage of cytochrome C and loss of mitochondrial membrane potential. Now, this programmed cell death or apoptosis, it can be induced by certain factors which can either be internal factors, for example, abnormalities in DNA during replication or during cell division, and they can be external stimuli like tumor necrotic factor alpha, etc. So the processes or pathways by which programmed cell death is achieved or apoptosis, it is divided into two pathways: an intrinsic pathway, which is a mitochondria dependent pathway, and an extrinsic pathway, which is a mitochondria independent pathway. Okay, so the main important these proteases which are involved in bringing about the autodestruction or the programmed cell death or apoptosis, they are called caspases. So what are caspases basically? Caspases, these are the ones that initiate and execute the process of apoptosis. So caspases are cysteine aspartate proteases, like they have a cysteine residue present at the active site of the enzyme, and they target an aspartate residue on the targets. So they are produced as inactive enzymes called the procaspases, and upon appropriate signals, the procaspases are cleaved to form the active caspases. So the caspases have this property, they are divided. The caspases, they are divided into two forms basically, the initiator caspase and the executioner caspases. So the initiator ones, they initiate the process of apoptosis, and the executioners, they bring about the programmed cell death. Now the executioner caspases, we have caspase 3, 6 and 7, which belong to the executioner caspases, each having a particular role in the process. So what basically are the targets of caspases? So they target protein kinases like FAK, which is the focal adhesion kinase. So this is very much important because inactivation of FAK or focal adhesion kinase results in the detachment of the apoptotic cell from its neighboring cells. It is a process by which collateral or localized damage to other cells is prevented. So apart from that, it attacks the cytoskeleton elements of the cell like actin, and the caspase also activates certain enzymes called as CAD, that is caspase activated DNases. So when these CADs, they are activated, they translocate from the cytoplasm to the nucleus, where they result in the fragmentation of the DNA. Another target of caspase is the lamins. We know lamins are present in the inner lining of the nuclear envelope. So when lamins are destroyed, it results in the shrinkage of nucleus and the disassembly of the nuclear envelope. So basically, when structural proteins, they are hydrolyzed, the nucleus breaks up, the cell shrinks and forms extensions. These are the extensions which are called blebs. So small membrane-bound fragments called apoptotic bodies, they bud off from these blebs and are engulfed by the phagocytic white blood cells. So the formation of blebs is very important because they prevent the leakage, because blebs, the apoptotic bodies, they remain membrane bound. So being membrane bound, it prevents the leakage of the cellular contents into the surrounding or to the other cells, so such that there is no collateral damage and no immune response whatsoever is elicited. Also, caspases, they have a few other functions, like they hydrolyze other structural proteins of cytoplasm as well, and activate DNA hydrolyzing enzymes. So this is a figure of the CAD, that is caspase activated DNA. So it basically, what it does, it cuts the DNA between the histones, resulting in DNA fragments of multiples. So if we subject a apoptotic cell to, if we subject this DNA to a technique like electrophoresis, we can see a ladder-like pattern, so which corresponds to the fragments of these DNA. Now, the pathways by which apoptosis is accomplished, as I said, it happens by two ways. One is the intrinsic pathway, which involves mitochondria or is mediated by the mitochondria, and the other is the extrinsic pathway, which is the receptor mediated. Now, first, the intrinsic pathway. So what happens in intrinsic pathway, whenever cell detects some damage, like a DNA damage or any other assault in the cells, what happens is that certain molecules, they get activated. For example, the genes of a family called BCL2 family. So we have this BAX gene or BAD gene. So when they get activated, they translocate from cytoplasm to the outer membrane of the mitochondria, where they are able to form pores in the mitochondria. So once these pores are formed in the mitochondria, it results in the leakage of the cytochrome C from mitochondria to the cytoplasm. So when this, which normally does not happen in a normal cell. So when cytochrome C is released into the cytoplasm, it associates with certain cytoplasmic factors like APAF1, which is the apoptotic promoting factor. So it reacts with APAF1 and results in the activation of the procaspases. So the procaspases, we know we have initiator caspases. So when initiator caspases get activated, for example caspase 9, it forms an apoptosome, which results in the activation of executioner caspases that finally lead to the apoptosis. Now in the extrinsic pathway, it is a receptor mediated pathway. So what happens, for example, we have taken the example of tumor necrotic factor here. So the binding of the TNF, which results while the cell is undergoing some abnormal changes. So the TNF, it binds to its receptor called the TNFR1, tumor necrotic factor receptor 1. So the binding of the TNF to TNF receptor induces a change, and the TNF receptor has a cytoplasmic domain which is called as the death domain. So these death domains, upon binding of the receptor, they undergo some conformational changes which activate certain adapter proteins, and these adapter proteins ultimately cause the activation of the procaspase 8. So the procaspase 8 is activated into caspase 8, which is the initiator caspase, and then the sequence of events goes until the executional procaspases are activated. Now both extrinsic and intrinsic pathway, though they are two different pathways, but these pathways, they converge at some point. Both the pathways, the mitochondrial and the activator mediated, they converge at a point wherein the effector caspases are activated. This point of conversion happens at the effector caspases, and then the downward process of the cell death is somewhat similar in both pathways, and both lead to the cell death ultimately. Now once this happens, so what happens, the cell, the apoptotic cell, as I said earlier, it has some peculiar morphological changes by which it is recognized by the phagocytic cells and is cleared up. So what are these changes? Basically, as I said, that it results in blabbing and formation of apoptotic bodies. So one important change here is the change in the a change in the membrane composition. So here the membrane does not break off or fragment, but there is change in the membrane composition. So what change is that? Basically, it is the change in the phospholipids. Normally in normal membranes, we have a phospholipid called phosphatidylserine. So phosphatidylserine is normally present in the inner leaflet, right? So but a cell undergoing apoptosis, what happens that this phosphatidylserine, it is flipped towards the outer membrane by an enzyme called scramblase. So when this phosphatidylserine is flipped outwards, it is it presents itself on the outer membrane of the cell undergoing the apoptosis. So this phosphatidylserine, it acts as a recruitment as well as attraction for a phagocyte. And the presence of phosphatidylserine, it acts as a signal called the find me signal. So ultimately this signal, it is recognized by the phagocytes, and the phagocytes, they engulf these apoptotic cells, and the process of degradation of the said cell continues. So in this manner, there is no leakage of the contents, and the cell, it has already lost its attachment with the neighboring cells. So there is no collateral damage to the other cells, and the cell is taken up and then taken care of by the phagocytes. So this completes the lecture on the apoptosis. Thank you for your time.